I'm Travis Block, the Chief Technology Officer at StemBioSys. And today I'm going to be presenting with Todd Herron, the CEO of CarTox, which is now a subsidiary of StemBioSys. And he's also the director of the cardiovascular core at the University of Michigan. And we'll be presenting on our technology, the CELLvo Matrix, and how we're using this extracellular matrix technology to solve the maturation problem associated with human-induced pluripotent stem cell-derived cardiomyocytes. So I'll be walking you through an overview of the CELLvo Matrix technology, and then taking you through how we developed and characterized our newest product, CELLvo Matrix Plus. And then Todd will be going into detail on the phenomenon we've observed by which iPSC-derived cardiomyocytes rapidly mature into cells that resemble those found in the human body when cultured on CELLvo Matrix Plus, and how this can be used to improve in vitro toxicity screening using these cells. So first for the matrix. So I always like to start not with the matrix, but what a cell looks like in its native niche. And so this is an image of a cell in its native microenvironment. And we're taught from a very young age in basic biology, before we got into more advanced stuff, that the cell is the basic building block of the human body. But in reality, if you could isolate every cell in the body and spin it down in a giant centrifuge, it wouldn't take up all that much space. In reality, we are composed mostly of extracellular matrix. And there is constant communication between the cells in our body and this extracellular matrix, as you can see in this image. Yet, typically, we ignore this. And actually, this image gets even more striking when, rather than looking at the outside of a cell in its environment, you look at a cross-section of a cell in its native microenvironment. And actually, until you label this image, it's actually very difficult to see where the cell ends and the extracellular matrix begins. And so what you're seeing here is actually the phospholipid bilayer with the cell on one side and the extracellular matrix on the other. And there is constant communication here that's going on by the extracellular matrix components actually interacting directly with cytoskeletal components. So really, it's kind of an arbitrary border between the cell and its matrix. Yet again, as I stated, we tend to ignore this using traditional culture methods. Today, in most cell culture, we isolate a tissue of interest, we dissect that tissue either enzymatically or mechanically to isolate cells of interest, and then we plate those cells into very simple culture environments. Often things like tissue culture plastic, or it could be a dish that's coated in fibronectin or collagen or laminin. And these cells immediately respond to the environment that they're put in by changing their gene expression, such that they often no longer resemble the cells that were found in the body. And this is a huge problem. And so over the last couple decades, more and more attention has been paid to how the microenvironment is regulating cell fate. And we've had a lot of interesting work done where people have looked at things like substrate stiffness, looked at the soluble factors that are in the extracellular matrix to see how those impact cell differentiation and cell phenotype and whether it's mature or immature. We've looked at cell-cell interactions where people are doing co-culture models, cell ECM interactions where people are looking at different types of binding sites, and then even morphology where, rather than looking at physical or chemical attributes, we're actually looking at the structure, the architecture of the ECM. And this study down here where we're looking at kind of structure and architecture of the matrix is actually one of the studies on cell matrix interactions that I find most interesting, because it shows how poorly we're able to approximate the microenvironment when we look at single variables. So in this study that I highlight here from PNAS, what they did is, taking a simple plastic substrate, they essentially made cookie cutter cutouts at the bottom in order to force cells to assume different morphologies. And by forcing the cells to assume different morphologies without altering the stiffness, without altering the chemistry of the substrate, without altering the number of binding sites, they were able to change gene expression and ultimately change differentiation pathways. And so this just goes to show that there's not one correct stiffness for cells for getting a desired phenotype. It's really going to be a complex interplay of all of these things. And so when people try to recreate a native niche by saying, okay, muscle has a certain stiffness, I'm going to give my substrate that stiffness using a biglycan gel and coat some fibronectin on top, and as long as I approximate that stiffness, I should be able to get the phenotype I want. That's really an overly reductionist approach that we try to avoid here. ECM physiologically is a complex mixture of structural and soluble proteins, and it's just not appropriate to try to recreate that using a reductionist model. And so what we do at StemBioSys is we actually take stromal cells from the body, we put those cells into cell culture dishes, we allow them to grow to confluence, and then we induce those cells to secrete a matrix. So essentially, we are growing a very thin layer of tissue at the bottom of the dish. And when we grow these from different tissues, they make distinct architectural, physical, and chemical properties based on the cells that were used to produce that matrix. And they elicit more biologically relevant phenotypes relative to traditional cell culture methods. And moreover, there's been a lot of innovations in the way people are growing cells these days. 3D cultures and organoid cultures have become more and more popular, but those processes are often very difficult to manufacture. The process that we're talking about today is very simple, scalable, and reproducible. And so often when I describe this, it's very difficult for people to get a feel for what I'm talking about. So the next slide is going to have some images of exactly what I'm talking about. So when you look up very close on the left side, this is an SEM image of the matrix. You're able to see that we have a coating of this matrix that totally coats the substrate. And when you zoom out a bit, this is a bright field image at 10x. You're able to see there's a fibrillar structure to the matrix, and we can visualize it in bright field. It's also anisotropic, so there's some natural alignment. And then it also, though we're getting a total coating of the matrix, it is transparent, and it does not interfere with bright field microscopy of your cells. So to sum up kind of the general overview of this CELLvo Matrix technology, these matrices create a natural protein-based environment for the isolation and culture of adherent cells. While they do this, while they coat the entire substrate, and while they are a very complex matrix with complex architecture, biochemistry, and physical properties, they're not complex to use. They fit into existing workflows. So when we sell these matrices, they come in dishes that look like normal tissue culture flasks. The matrix is totally transparent, and so you actually can't tell it's even there until you look at it under a microscope. And these matrices are compatible with fluorescent imaging. I think you may see some more images like this in Todd's half of the talk. He happens to be a beautiful microscopist. And then these cells have a more native phenotype when cultured on CELLvo Matrix. So one thing that we've observed is that when people put cells onto more traditional, more simple substrates, cells tend to immediately upregulate matricellular proteins that aren't in their environment. So if you put cells, say, onto a fibronectin substrate and they're expecting to see collagen 1 or collagen 6, they're going to immediately upregulate all these proteins that they expect to see in their native niche that aren't present. And so for the gene expression of cells, the matrix components that are not present can be just as important as the ones that are present. And so this is more evidence as to why we think that it's important to more closely replicate the physiologic niche of these cells rather than taking on these more reductionist models. So in short, we're enabling biologically relevant cell culture. But while I'm talking about recreating the native niche, it's important to note that there's actually a whole lot of different niches in the body, and that not all cells are requiring the same niche to grow and behave normally. And so this is a paper that came out of our scientific founders lab, Dr. Chen, that I worked on a bit during grad school with Dr. Marinkovich. And so the key findings of this paper were that cells from different tissues would elaborate matrices that retain properties specific to the tissue source. So in this paper, we took bone marrow and adipose-derived mesenchymal stem cells, and using the exact same process, induced them to secrete a matrix. And when we looked at the composition, perhaps the most interesting finding wasn't the differences in the matrices, but rather the similarities. MSCs from adipose and bone marrow tissue made matrices that were extremely similar in terms of the biochemical composition. But when we looked at the structural and physical properties, they were extremely different. So the bone marrow matrix made a much stiffer matrix. They were both rough compared to TCP, but the bone marrow matrix was rougher. And it was also much more aligned and more anisotropic. And so not only, I'll get more into this more in the next slide, but not only did they make matrices that retain properties specific to the source tissue, but also tissue-specific extracellular matrices influence cells in a predictable manner based on the source of cells used to make the matrix. And then these tissue-specific properties were really critical for determining cell fate in vitro. And this could, have implications for in vitro screening applications if we're able to get more biologically relevant phenotypes. So now we've applied this model that was created in this paper and validated it in this approach in multiple systems. So on the left, this is data actually from that same paper, where using the bone marrow matrix versus the adipose matrix, we were able to drive differentiation of mesenchymal stem cells down an osteogenic or an adipogenic pathway. And so, based on the source tissue, the adipose matrix, the ADECM, promoted adipogenic differentiation, whereas the bone marrow-derived matrix promoted osteogenic differentiation. And so, despite similarities in the composition, they made different structural and mechanical properties and induced different function. Later, in a paper published last year, we showed that the same system could be applied to an articular chondrocyte-derived matrix, where a matrix produced by articular chondrocytes promoted greater chondrogenesis relative to either tissue culture plastic, which is the standard method for expanding chondrocytes, or a bone marrow-derived matrix. But one of the most interesting things again here is that the bone marrow-derived matrix was still vastly superior to tissue culture plastic in maintaining the gene expression of chondrocytes. And so this suggests a role both for tissue specificity of matrices, but also it suggests that common motifs exist in these extracellular matrices. So it's possible that one matrix, while optimal for a certain cell type, may work well for preserving phenotype of a wide array of cells. And so it may not be that you need a different matrix for every single cell type in the body. It may be that some combination of matrices could be good enough to approximate a much wider array of in vivo niches. And so with that, I'll move on to describing CELLvo Matrix Plus. And so in our lab, we produced a matrix. Our original product, CELLvo Matrix, is produced by bone marrow-derived mesenchymal stem cells. And we recently produced the CELLvo Matrix Plus from amniotic fluid-derived stem cells. Each of these matrices are fibrous and anisotropic, but they have a lot of differences in function and composition. And so the first thing we did with this was we tested Matrix Plus in a variety of applications to see if it had any interesting value worth investigating further. So the Matrix Plus improves the differentiation of mesenchymal stem cells. Here, this is looking at the culture of elderly adipose-derived mesenchymal stem cells at a very late passage. These are passage 9 cells, and culture on Matrix Plus improved proliferation of these cells and allowed them to differentiate more efficiently. It also supports the self-renewal of induced pluripotent stem cells in a variety of media. So relative to competitive products, Matrigel was tested here, we were able to support self-renewal of iPSCs on par with that product. It also supports self-renewal of MSCs during extensive passaging. So I just mentioned that these were passage 9 cells. We're able to rapidly expand mesenchymal stem cells without loss of function. And then this last observation here is what the rest of this talk is going to be about, is that we observed that on Matrix Plus, iPSC-derived cardiomyocytes rapidly and spontaneously matured. And so this image on top here, this is an iPSC-derived cardiomyocyte on Matrigel. On the bottom, this is one on Matrix Plus. And so why is that important? It's important because iPSC-derived cardiomyocytes do not resemble mature cells. Sometimes this is referred to as Peter Pan syndrome. We can differentiate the cells and get them to a certain state of maturity that is fetal-like, and they never mature past that to where they actually resemble those cells found in mature adult humans. And so this is taken from a review paper in 2018, summarizing many of the differences between iPSC-derived cardiomyocytes and adult cardiomyocytes that Todd is going to go into more detail on later. But the key takeaway that I want you to have here is more general. It's that in theory, pluripotent stem cells can produce any type of cell in the body, but in practice, it is very seldom that we are able to achieve mature phenotypes that resemble cells found in adult humans. Instead, we say that pluripotent stem cells can make any type of cell, but really we can make any type of cell-ish. Using established differentiation protocols, we're able to get, beating cells that resemble cardiomyocytes, but don't have the complexity of ion channels that adult cardiomyocytes have. We're able to make islet cells that don't respond to glucose. We're able to make neurons that don't fire in the same way or don't express the same proteins. And so for many cell types, this is an issue. And this is a major problem for drug and toxicity screening applications, where very nuanced differences in phenotype can be critical to determining cell responses. So in the case of cardiomyocytes, for instance, if your cell is dominated by only the hERG ion channel receptor and you have a compound that blocks the hERG ion channel, it's going to appear cardiotoxic, even if in vivo it would not be. So we have a problem. That problem is that iPSC-derived cardiomyocytes are immature and fetal-like, and this is a major limitation to their widespread use for drug and toxicity testing. On the left, we have what these cells look like on Matrigel, which is a standard substrate for culturing these cells. And on the right, we have what these cells look like in the body. On the right, they're tightly packed, aligned, and rod-shaped. On the left, they are basically totally randomly oriented and totally round. So we have a hypothesis that traditional cell culture approaches lack critical extracellular matrix cues that are necessary for cardiomyocyte maturation, and that an appropriate microenvironment will support iPSC-derived cardiomyocyte maturation. So from there, again, Todd is going to go into detail on what we've observed with cardiomyocyte maturation, but I'm going to give a little bit more detail on our characterization of Matrix Plus relative to CELLvo Matrix, because that same phenomenon of taking on a mature morphology was not observed with CELLvo Matrix. And so I think it's an interesting comparison. It gives context. And so again, both of these matrices are fibular and anisotropic. However, the CELLvo Matrix appears to be more tightly organized relative to Matrix Plus. In terms of the physical properties, Matrix Plus is more adhesive. It's a smoother matrix, so it's both flatter and has less roughness in terms of ridges of the fibers. And then Matrix Plus is much less stiff, on the orders of 2 orders of magnitude less stiff relative to CELLvo Matrix. And this is interesting because all of these differences have been discussed in the literature as things that can contribute to cell phenotype, and especially stiffness has been extensively studied in relation to cardiomyocyte maturation, with softer matrices being shown to promote more cardiomyocyte maturation. However, on the substrate, we're seeing cardiomyocyte maturation way in excess of other substrates that are soft. And so there's something else going on there. And those physical differences must be underlied by biochemical differences. It must be a difference in composition. And so when we look at mass spec, we see that in this heat map, all of the columns are different donors. On the right, we have 3 donors of bone marrow that were used to make CELLvo Matrix, as the CM1, 2, and this should be 3 right here. And then on the left, we have Matrix Plus, MP1, 2, and 3. Those are 3 donors of each. We used hierarchical clustering to organize these. So we see that based on composition, they naturally cluster, such that the differences between Matrix Plus and CELLvo Matrix are greater than the differences between donors. And then we see that there's differences both in the classes of proteins that are present, with Matrix Plus being relatively proteoglycan rich and collagen poor relative to CELLvo Matrix. And we also see some interesting individual differences. So Matrix Plus, it totally lacks collagen 6. We were very surprised by that because many progenitor cell niches in the body are rich in collagen 6. And that's the most abundant protein in CELLvo Matrix. Whereas Matrix Plus is much richer in heparan sulfate proteoglycan, also known as perlecan, relative to CELLvo Matrix. This is interesting because mutations in that protein can cause either in utero death or death shortly after birth due to cardiac malformations. So it's potential that it's possible that this protein is critical to the maturation process that we're observing here. But again, as I observed in the adipose versus bone marrow derived matrix study, one of the most interesting things to me is how similar these 2 matrices are. Actually, roughly 70% of the composition is shared between the 2 matrices, even though we see very different phenomena on the 2. So I'm just going to give a brief overview of the Matrix Plus characterization before I hand it off to Todd to go into detail on the cardiomyocyte maturation. So first, there's the consistency. This image right here is principal component analysis, with these 3 clustered dots being the 3 batches of Matrix Plus, where these others are 3 batches of CELLvo Matrix. And so the close clustering shows very consistent product independent of the donor used to produce the matrix. And then the stiffness. I mentioned before that there's an abundance of literature discussing the role of stiffness in cardiomyocyte maturation. And so it's interesting that the CELLvo Matrix Plus is so much less stiff than CELLvo Matrix. In terms of composition, we want to investigate further whether or not there could be a mechanistic role for the perlecan in Matrix Plus, because of the fact that mutations in this protein can cause issues with cardiac formation in vivo. However, while it's tempting to look at these interesting correlations and interesting presence or absence of components, interesting coincidences between what we're observing here and what's been reported in the literature, it's tempting to think that we can identify a mechanism or a potential mechanism and recreate this phenomenon with an artificial substrate, some kind of hydrogel that we're coating in fibronectin or collagen to help cells stick to it, and recreate the same phenomenon. But I want to reiterate that the ECM is a physiologically complex environment, and it's just not appropriate to think that we can recreate that using just a couple of proteins. It's more likely here that there is an entourage effect where the cumulative effect of all of these matrix properties interacting with the cell is actually greater than the sum of any one of these properties, or any combination of these properties. And with that, I'm going to turn it over to Todd.
All right, thanks, Travis. I'm going to share my screen. Let's see.
You're going to swap it.
Okay. All right, so you can see my screen now.
Yes, sir.
All right, thank you, Travis. That was a great introduction to the importance of the extracellular matrix for cell physiology and for cell culture. And now I'm going to show you data that we've collected over the last 3 years that has really convinced us that the Matrix Plus matrix is an optimal matrix to promote human cardiomyocyte, human heart cell maturation. So all of the testing that we've done has been done using commercially available cardiomyocytes, which are available from Cellular Dynamics International. These cells are optimal because they provide a standardized product of human cells that are quality controlled for human cellular assays. Working with these cells is very convenient. The cells are cryopreserved, so we can really control the rate at which we plate the cells and make discoveries. And importantly, this cell type has been validated by international FDA-led validation studies. And now the use of these cells actually can be included in investigational new drug applications with the FDA. So when StemBioSys approached me and asked if I would test the effects of their ECMs on cardiomyocytes, this is the protocol that we came up with. So we start with a vial of cryopreserved cells, and then that batch of cells is divided into 3 different experimental groups. In one group, we plated cells on a mouse-derived complex matrix called Matrigel, which we use routinely in my laboratory, and we have published that this Matrigel complex ECM does promote some maturation of cardiomyocytes. We also used another matrix that Travis introduced called CELLvo Matrix, was another experimental group. And then finally, we tested Matrix Plus ECM, where we coded from the same batch of cells, IPS confluent monolayers in 6-well dishes. And then we did some basic experiments. We did some basic microscopy to look at cardiomyocyte structure and to look for expression of some markers of cardiomyocyte maturation. And we also did some pretty extensive functional studies where we performed electrophysiology screening and medication screening to look at the utility of ECMs for high throughput screening and pro-arrhythmia assays. So this is a phase contrast image from a microscope of IPS cardiomyocytes cultured on our standard Matrigel matrix using our standard protocols. So this is a fairly large field of view, and you can see that the cells, so where the very dark spots are, those are the nuclei of each cell, and you can see the cells are circular, and they have very random shapes from cell to cell. When we looked at the cells cultured on CELLvo Matrix, which is a bone marrow cell-derived human matrix, we saw a cell morphology that was very similar to Matrigel. So there was not any promotion of maturation there. But when we looked at the cells cultured on the new Matrix Plus matrix, we could very quickly observe differences in the cell morphology, where we can see that the cells are much longer and rod-shaped in nature. And this was very quick because it was just 7 days after we thawed the cells from cryopreservation. So this led us to do more imaging and to do more studies. And this shows very nicely the impact of the different ECMs on human cardiomyocyte morphology. So on the left here, you can see human cardiomyocytes plated on the mouse-derived matrix, matrigel. So you can see the cells are circular, and they don't have really any orientation to them. On the other hand, the same cells from the same vial of cells cultured on Matrix Plus, you can see profound differences in the cell morphology, where nearly 100% of the cells become rod-shaped and start to take on some orientation relative to each other that is much more adult cardiomyocyte looking. We were very excited to be able to see sarcomeres of the cardiomyocytes just by phase contrast imaging. So here, this is a picture of a human cardiomyocyte. All of these rod-shaped cells are cardiomyocytes, and you can actually see striated pattern within the cell. This is what a cardiomyocyte looks like if you obtain and do a digestion of the heart. You would see adult myocytes that look just like this with this repeating light dark pattern. And until we used Matrix Plus over the last 10 years of culturing these cells, until we used Matrix Plus, we never observed sarcomeres that were visible by phase contrast. So that was very exciting to be able to see that. So important things to point out again is that this change and this maturation of cell morphology is occurring just within a week after thawing these cells from the liquid nitrogen cryo storage. So it's a very rapid effect. Importantly, as Travis pointed out, and you can see here, even though there is a coating on the plate, the coating does not interfere with microscopy. So you can still obtain very good images with standard microscopy. And I don't think Travis touched on it, but another very nice feature about these matrix coated plates from StemBioSys is that the plate is ready to use upon arrival. There's no need to do like a matrigel coating where you have to work on ice, you have to do a complex coating of ECM on the plates. The plates are already coated and ready to use. Another feature that we noticed is that, as we commonly see with matrigel, the mouse-derived extracellular matrix, the cells do not take on any alignment or any directionality. On the other hand, with Matrix Plus, looking over a large field of view, you can start to see fiber tracks that are being formed, where the cardiomyocytes are lining up relative to each other and creating bundles of fibers that work together to generate tension and perform the function of the human cardiac organ. So here, this is commonly referred to as an isotropic organization of the cardiomyocytes, but with Matrix Plus, what we see and what is commonly referred to in the whole heart is anisotropic organization of the cells, where they have some directionality to them. Next, we did some functional testing of the 3 different matrices. Here we did an electrophysiology assay using voltage-sensitive dyes. So on the left, you can see for 3 different matrices. The top one is for matrigel, and the middle is for CELLvo Matrix, and Matrix Plus is on the bottom. These are spontaneous pacemaker action potentials that the monolayers generate themselves. So these cells, when you culture them together, they will create a little piece of tissue which has a pacemaker, just like your heart has a pacemaker in it. And then we did drug testing. So the common ion channel that's used to screen for safety of compounds is called the Hurg ion channel. So in this assay, we applied a Hurg ion channel blocker. And what we saw in matrigel is a typical response. We saw that the action potential duration, the width of each of these contractions got wider, which can lead to arrhythmias. On CELLvo Matrix, we saw something similar, even though the effect was much more exaggerated with very extreme action potential duration prolongation. But with Matrix Plus, we saw something interesting. With the same dose of the Hurg channel blocker, we could actually see the occurrence of what's called TDP arrhythmias. TDP stands for torsade de pointe, and this is the fatal arrhythmia that drugs can induce that the FDA and drug makers are trying to avoid. So here we show that we can actually observe drug-induced arrhythmia in a dish if we use Matrix Plus. And here I just want to show you an example of what that experiment looks like. So here you can see a monolayer of human stem cell-derived cardiomyocytes. The pacemaker site is right around here. And you can see very nicely that the action potential or the electrical signal spreads very nicely through the monolayer, traveling very quickly through the monolayer. And what we observed when we added the hERG channel blocker is we observed that normal propagation turn into a stable reentrant arrhythmia, where the activation doesn't seem to start at any one point, but is stuck in a reentrant loop. This is what happens to the action potential activation in the heart during ventricular fibrillation. So this type of activation pattern in a rotating wave precludes force development and ejection of blood from the heart. So we were very thrilled to be able to see that we could induce torsade de pointe in a dish using a drug. And now we can go beyond what typically is used in the field for cardiotoxicity is only a surrogate for an arrhythmia, but here we can actually show an arrhythmia. This time recording shows the progression of the E4031 effect. So here you can see a normal action potential. At this point in time, we added the hERG channel blocker. And then by the fourth beat, we could start to observe prolongation of the action potential. Then we could start to observe early after depolarizations. And then these actually degraded into TDP-like arrhythmia. So next we went on to create high throughput screening plates, 96 well plates coated with Matrix Plus in order to do some medication screening in high throughput. So again, using the voltage sensitive dye, this is what the readout looks like. So this movie on the left is a 6x6 recording of a 96 well plate. And each monolayer has spontaneous activations. And doing the experiment in this type of high throughput screening assay, we can generate dose response curves quickly, and we can look for the effect of medications very rapidly. To do some validation, here we show for 7 different 96 well plates, the average spontaneous beat rate. You can think of this as similar to the heart rate, only here this is shown in hertz. So the average beat rate is around 1 hertz for the 7 different plates. 1 hertz corresponds to a heart rate of about 60 beats per minute, which is similar to the normal adult resting heart rates. Maybe on the low side, but for an athlete, maybe 60 beats per minute would be normal. Then another important variable for high throughput screens is the variance within a plate and also the variance between plates. And so here we have calculated the variance of beat rates per plate. And for every plate, we see that the variance is well below 6%, which is compatible with meaningful high throughput screening assays. When we did medication screening, we used a panel of compounds that have been approved by the HESI Cardiac Safety Committee, which is led by the FDA. And these compounds are called CIPA pro-arrhythmia compounds. CIPA is a movement by the drug discovery field and the FDA to improve the cardiotoxicity screening of drugs before they become medications. And so for 6 different drugs, I have plotted here the type of arrhythmia that we observed at the highest dose of each of these compounds. So a drug called domperidone is known to have some risk to cause arrhythmias, and we could detect tachyarrhythmia or torsade de pointe with this drug. Another high-risk compound called disopyramide, which is used clinically, but at high doses can cause arrhythmias. We could observe, again, action potential prolongation and the occurrence of early after depolarizations. Another compound called azimilide is another antiarrhythmic compound that can cause arrhythmias. And here we could detect arrhythmias at a high dose of azimilide. Similarly, with sotalol, we could observe arrhythmias. With ibutylide, we could see similar APD prolongation and the occurrence of extra depolarizations. And importantly, for a calcium channel blocker called Bepredil, we observed total quiescence with this calcium channel blocker at the highest dose of Bepredil tested. And all the dosing that we did was based on recent publication from the FDA and Blanova et al., which was published in Cell Reports. We did compare the outcome of the high throughput screening that we did on Matrix Plus to some historical data that we have where we tested a subset of those compounds with Matrigel. And what you can see for each drug, which is listed in the bottom, the occurrence of arrhythmias. So for each drug, we tested 6 different wells with the same dose of the drug. And you can see a lot more red with Matrix Plus, which indicates that for the high risk compounds at this submaximal dose, we observed many more arrhythmias when cells were cultured on Matrix Plus relative to the same compounds being tested on Matrigel, which indicates that Matrix Plus provides a higher sensitivity electrophysiological pro-arrhythmia assay than the other ECM that we tested of Matrigel. Importantly, though, if you look at the low-risk compounds here where the bars are mostly green, even though Matrix Plus provided a high sensitivity assay, we also feel that it's very specific because the low-risk compounds, such as ranolazine, did not induce any arrhythmias at any doses. So we feel that Matrix Plus provides a very, very good ECM substrate to perform human cardiotoxicity screening on. So then we've gone on to perform more structural analysis since we observed these effects of maturation. So here you can see immunostaining and confocal images of cardiomyocytes that were cultured on Matrigel in the left column. Compared to the same batch of myocytes cultured on CELLvo Matrix Plus. So here in panel A is staining for troponin I, which is one of the myofilament proteins involved in contraction of the heart. And on Matrigel, you can see a radial organization of this sarcomere protein. But on the other hand, with Matrix Plus, we see sarcomeres that are in register with each other and are uniformly organized throughout the length of the cell, which is much more rod-shaped. And also I should point out that the cells on Matrix Plus are much more often binucleated, which is an important hallmark of maturation of the heart. So in fetal development, in the developing heart, the cells have one nucleus, and then shortly after birth, in the hearts, the cells become binucleated because the nucleus starts to divide without the cell dividing as the heart matures. So to be able to see binucleated cells also is very exciting and also a very good indication of maturation of the cardiomyocytes. So when we look at staining for cardiac troponin T, which is another contractile protein, which works in concert with troponin I, with cells cultured on Matrigel, again, the organization is radial, but with CELLvo Matrix Plus, the sarcomeres are beautifully aligned relative to each other in a way that will promote better force production than in a circular, radially organized cell. Similarly, with alpha-actinin, another component of the sarcomere of the heart, responsible for the contraction, you can see radial organization on matrigel, which becomes rod-shaped and much more organized when cells are cultured on Matrix Plus. So finally here, the morphology quantification. We calculated the circularity index of cells cultured on matrigel compared to CELLvo Matrix Plus. And you can see that the cell circularity index is much lower for cells cultured on Matrix Plus, which shows a very robust indication of the impact of the cell matrix on cell morphology. So next here I'm showing monolayer pictures where the density of the plating is a little bit higher to form monolayers. So the panels A, B, C, and D show human cardiac monolayers cultured on matrigel. And on the right, panels E, F, G, and H. These are for cells cultured on Matrix Plus. DAPI stains the nucleus, so you can see the cells are there. The green stain is called phalloidin. Phalloidin stains the cytoskeleton, the inside of the cell, and shows you how the cytoskeletal architecture looks inside the cells. And it's very apparent that on matrigel, the cytoskeleton is disorganized and has random orientations going in all directions. But if we look at the phalloidin staining using Matrix Plus, we can see that there's actually a predominant vector where the cytoskeleton is actually organized from cell to cell. And this makes me think of the image that Travis showed at the beginning, where if you look closely, you can't tell where the ECM begins and the cell begins. So this shows that the matrix can help promote organization, not just within one cell, but how cells organize with each other to create tissue or an organ. So another important feature from this slide is looking at the red staining. So the red staining here is for a protein called cardiac troponin I. Cardiac troponin I is a critical marker well known to indicate maturation of human cardiac muscle. And we can see with matrigel, there are some cells that do express cardiac troponin I, but there also are very many cells that do not express any cardiac troponin I. So there's a heterogeneous cell mixture there. However, on Matrix Plus ECM coding, the cardiac troponin I signal is present in nearly every cell that we image, which indicates that there's a more uniform maturation happening and much more extensive maturation occurring when cells are cultured on Matrix Plus. And we think this is really important for future development of heart medications because cardiac troponin I is actually used clinically as a blood biomarker of heart damage. So when there's a heart attack or there's any damage to the heart, cardiac troponin I levels will rise in the blood, and cardiologists use that as an indication that there may be some cardiac damage. So to be able to see robust CTNI expression is very good for the future development of cardiotoxicity assays. So we've also, in addition to looking at the muscle protein structure within the heart muscle cells cultured in these matrices, we've also started looking at the distribution and function of the energy suppliers within the heart. So within the heart, the mitochondria are the intracellular organelle responsible for producing the bulk of the energy used for every heartbeat over the lifetime of a person. And so here we've used a fluorescent dye called MitoTracker red, and then some chemical acronyms, CMX-ROS. So this dye will actually go into living cells and will accumulate in the mitochondria. And it accumulates mostly in mitochondria that are functioning at a very high level. So the more red you see, the more functional the mitochondria are. And what you can see, just looking at comparing panel A to panel B, which is comparing matrigel ECM to CELLvo Matrix ECM, we can see that in panel B, the red signal is much more apparent. It's much brighter. When we do higher resolution imaging using a confocal microscope, here in panels C through E, we can see that with Matrigel, if we look at a single cell here, for example, most of the mitochondrial signal is accumulating around the nucleus rather than in the cytosol where the muscle proteins are that need energy. If we look at the same cells cultured now on Matrix Plus and look at the mitochondrial distribution, we can see two things. One, we can see much brighter fluorescence throughout the cell. And this indicates that not only are there more mitochondria distributed throughout the cell, but also those mitochondria are functioning or capable of functioning at a much higher level than the fetal cells on Matrigel. And then finally in panel E, we obtained adult ventricular cardiomyocytes from a mouse and put those into culture and did the same mitochondria staining. And so you can see here, this is a normal adult cardiomyocyte and what it looks like when you stain the mitochondria. And when we do the quantification matches the images where Matrigel shows the lowest mitochondrial mitotracker signal. Cells cultured on CELLvo Matrix Plus have a mitotracker signal that's much more similar to the adult cardiomyocyte mitochondrial signal. So this shows that Matrix Plus promotes mitochondrial distribution throughout the human cardiomyocytes, and also that the mitochondria membrane potentials are hyperpolarized and able to work at a higher capacity when cultured on Matrix Plus. And importantly, the Matrix Plus signal is approaching the levels of the adult ventricular cardiomyocyte. So for summary and conclusion regarding the effect of Matrix Plus to promote maturation, it's very exciting that Matrix Plus offers a human ECM with properties that will mature cryopreserved cardiomyocytes in a very short window of 7 days. I have to say that this is a major advance over other currently used protocols, which require sometimes a month long of cell culture with complex electrical pacing or complex addition and removal of hormones or other medias. This is a very straightforward maturation method where cells are simply plated on a matrix and the cells become mature in a short period of time. Importantly, I've shown that Matrix Plus induces cardiac troponin I expression. This is very important. Cardiac troponin I is a well-known marker of cardiac maturation. And as I pointed out before, the expression of cardiac troponin I has implications for future biomarker detection in cardiotoxicity assays. For example, in the Matrigel coding where cardiac troponin I levels are very low, if we were to use CTNI as a marker for damage, we might not see any signal at all because the cells do not express cardiac troponin I. However, with Matrix Plus, we can produce cells that express proteins in a similar way to the adult heart, and we can use clinically relevant biomarkers in vitro to detect cardiotoxicity. And we also think it's very important the effects that Matrix Plus has on the mitochondrial maturation. The mitochondria is the energy supplier of the cell, and so to make good, meaningful cardiomyocytes, we need lots of mitochondria functioning at a very high capacity. And we think also being able to produce cells with much more robust mitochondrial function will have implications for future cardiotoxicity testing, and making the cardiotoxicity screening more sensitive and also more specific. And so that's the conclusion. If there's any questions, we're happy to take questions either through the chat window or through unmuting yourself.
So we actually have a number of questions that have come in through the chat window. So I'll start by reading off a few of these and either take them myself, or I think a lot of these will be better addressed by Todd. And if you have any other questions, you can add them to bottom of that. And just before I get into the questions, I want to mention that CELLvo Matrix Plus has been launched commercially by StemBioSys, and so you can buy it through www.stembiosys.com. You can also email us at info@stembiosys.com with any more questions, and we're always open to new collaborations as well. So the first question was, I would like to know more about the lot-to-lot, donor-to-donor reproducibility of the Matrix Plus. It's somewhat encouraging to see that in 2D PCA, the Matrix Plus segregates from the regular matrix. But in the mass spec analysis, it looked like substantial, greater than twofold variation in key protein levels between lots of Matrix Plus. Do you have some functional screening as part of your QC, similar to how lots of Matrigel are qualified, to determine whether a lot is within spec or not? So I'll go ahead and take that one. As far as the mass spec data that we showed today, for ease of visualization, individual components were normalized independently of one another. This was done because otherwise the most abundant proteins would be very darkly colored and it would be impossible to differentiate the others. And so it's just for ease of visualization. This is all data though that's being published, and both in a pending manuscript that's under review as well as in our patents. And so we're happy to share more data on the composition of these matrices. And so the only proteins where we were seeing something around twofold or greater variation within one of the matrix products were ones that were at very low abundance. So something that was 0.1% composition in one lot might be 0.2% composition in another lot. But the most abundant proteins were always the most abundant proteins and in similar ratios. And then as far as a functional screening as part of our QC, we do have functional screening. In fact, that's our key lot pass criteria. However, currently our functional QC is not related to cardiomyocytes. When we were initially developing the product, we had other applications in mind before we discovered how well it worked for cardiomyocyte maturation. So we have functional QC, but it's not currently related to cardiomyocyte maturation. And then in discussing lot-to-lot and donor-to-donor reproducibility, it's also, I think, important to mention that from a single donor, we're able to produce thousands of plates of matrix. And so if somebody wanted to ensure a supply of consistent matrix, we'd be able to produce thousands or hundreds of plates for somebody from a single donor. And then that also allows us to QC new lots based on that to ensure reproducibility. The next question is one that I'll address to Todd. Why were EADs not observed in Matrigel control for 500 nanomolar of E4031? EADs with eye cells have been previously reported consistently starting at 30 nanomolar.
All right, so we have observed EADs at 500 nanomolar. Just in that example, there weren't EADs. So when we use E4031, typically with Matrigel and other coatings, we see a wide range of responses ranging from extreme prolongation without EADs to prolongation with EADs, and sometimes even tachyarrhythmia. So the EADs are there, just in the examples that I showed, they didn't have EADs.
Okay, next question. Can you elaborate on the differences in your control Matrigel system compared to standard SIPA studies? In the Blanova 2018 paper, arrhythmias were observed in nearly all, if not all, of those high-risk compounds, and many of the intermediate risk ones that you showed have only normal rhythms in your, and many of the, okay, so the Blanova paper had arrhythmias in nearly all of the high-risk compounds and many of the intermediate ones, though they only had normal rhythms in the data that we showed.
Right, so I think that's the data that we showed with the red and the green. That was for a sub-maximal dose, which is just at 10 times the effective therapeutic plasma concentration. So for those compounds, we did observe action potential prolongation at those submaximal doses. And on Matrigel, at the higher dose, which is oftentimes 100 times the ETPC on Matrigel, we did observe arrhythmias just like in the Blanova paper. So I think maybe it wasn't clear that that graph was for its submaximal doses.
I think, and I don't want to be mistaken here, but I believe in the Blanova paper, some of the data on 10x ETPC can be found in the supplemental data. But I could be mixing it up with another one of the FDA studies.
Mm-hmm.
The next question, how do CELLvo and Matrix Plus compare to decellularized matrix from competitors? Todd, if you don't have anything to add on this, I can speak a little bit in general terms, but I haven't tested cardiomyocytes.
We haven't done that kind of testing.
So the only thing I can speak to in more general terms on that is that we like the fact that we're able to decellularize this and maintain structure. And so I alluded to the anisotropy of these matrices that may be a driver of how these cells are aligning. And so when you are with these decellularized matrix products, typically it's ground up tissue that's turned into a powder that can be resuspended and then coated onto a dish. And so there's an additional resuspension step, an additional coating step, and we're losing that structure. So I can comment definitively that this is going to be easier than doing some of that, unless there's some of those that are pre-coded that I'm not familiar with. But in terms of the function, we have not tested those. The next question, could Matrix Plus be used as a standalone matrix product, just like commercial Matrigel, for coating on other specialized cell culture substrates or 3D tissue engineering application? So the shortest answer to this is that we don't know yet. This is something that we've investigated briefly in the past, not with Matrix Plus, but with our original product, and was not a priority at the time. It's possible that either the structural or physical properties that we're generating are critical. It's possible that they're not. And so it's something that we could investigate, but we don't know the answer to now. The next question, I believe, Todd, okay, you brought up this answer already. How long do you have to culture on Matrix Plus to achieve the elongation and binucleation you demonstrated in cardiomyocytes?
Yeah, so all the imaging that we showed was on day 7 after thaw. We have done time-lapse imaging from the time of plating up to 7 days, and we can start to see the divergence of cell morphology after about 4 days. And then by day 7, the cells look like you saw in the presentation here.
Is it possible to put Matrix Plus onto assay systems that allow electrical pacing of the iPSC CMs, e.g. ASEA, Axion, Nanlon, MEA plates? Have you done any tests for responses to positive ionotropic compounds or looking for a positive force frequency relationship?
We have had interest from MEA plate users, and at StemBioSys they have done some custom plating of Matrix Plus onto, I believe, all of these kinds of plates. And in our lab, we have tested only the Axion plates, and there we did do some recordings where we could pace the cells through some of the electrodes in the dish, and the coating seemed to stick pretty well and be feasible for the MEA plate type of assay. But we didn't do any force frequency responses because at the University of Michigan, our Axion instrument is a bit dated and it doesn't have those modalities to do contraction measurements.
And I'll just add to that coating these plates is something that we are doing on a custom basis now with collaborators. However, it's not something that we currently offer as a standard product. Anytime we move to a new format, it requires some new optimization work. Actually, this was something I was looking at just today before this call. And so this is certainly something that we plan to put additional effort into to standardize and make available. And the last question that we have currently in the chat page, it's Todd mentioned that all experiments over the last 3 years were with iCells. Have you tested any other iPSC-CM cell lines, or is it possible that this is a this maturation is somehow donor specific to the iCells?
Yes, we don't know the answer to that. We've only tested the iCellmyocytes, just because that's the cell type that's used most widely for drug discovery and toxicity testing. We have used other disease cells from cellular dynamics and could see disease phenotypes. So yeah, the answer is we don't know for sure yet. We do need to test other cells as well.
Excellent. There don't appear to be any additional questions. We can hang out on here for a few more minutes, but with that, I will thank everyone for tuning in and remind you to follow up with us if you have any questions. Thank you all.
Yep, thank you.